H plus. Okay, this H plus is indicating that it is in the additional proton is in the solution. Okay, or sometimes you might have also seen writing it like this, NADH2 also some books write, okay, or NADH, but I will be using this one NADH because it is more convenient for me. Okay, so these are some of the basic concepts. So NADH is going to get oxidized. Okay, similarly FADH2 is also going to get oxidized. Okay, or FADH is also going to get oxidized, or FADH2 is also going to get oxidized. Okay, now these two are going to get oxidized. NADH and FADH2 are going to get oxidized. Now we will see the what are the complexes and how they are working. Okay, now yeah, so this is a schematic representation of we will just see the complexes. Now these are the complexes. Now this blue color lines that I've shown here, this is the inner membrane. Okay, when I'm drawing two lines, don't get confused because membrane are bilayered. So lipid molecules, let's assume these are the lipid molecules here. Okay, so these are the lipid molecules, something like this. Okay, so because it is a bilayered membrane, I have drawn two lines. Okay, now on these bilayer membrane we have the complexes. We have the complexes on this what layer membrane? On this bilayered membrane we are having the complexes. Okay, now these complexes how many complexes we have in total? We have 4 complexes in total. Okay, for electron transport chain we have involvement of 4 complexes. Now if you see the NCRT, all the 4 complexes are shown to be transmembrane complexes. What are transmembrane complexes? Transmembrane complexes means, see here, the complex is traversing the membrane. A part of the complex is outside, a part of the complex is embedded within the membrane. So such complex are transmembrane complex. All the 4 are shown like that. But if you see in real, I mean in some good books, okay, this complex 2 is not a transmembrane complex, but still in the NCRT whatever is given we should follow that for Neet, but I am telling you the correct one. Okay, now but still when the examination you should still follow what is given in the NCRT only. Okay, now this is the complex 1. Okay, so what is the name of complex 1? The name of complex 1 is NADH dehydrogenase. Okay, the name of the complex 1 is NADH dehydrogenase. Okay, NADH dehydrogenase. You have to remember this. Okay, NADH dehydrogenase. If you say it 5 times, you'll get it, no issues. So the first complex is NADH dehydrogenase. So in this complex 1, okay, we have FMN, flavin mononucleotide, and a iron sulfur center. Okay, we call it an iron sulfur cluster or an iron sulfur center. So this is complex 1. So what is going to happen at complex 1? So complex 1, whatever NADH that are produced in the glycolysis step, okay, we have shuttles, malate aspartate shuttle. Okay, these shuttles will decide whether the electrons from the glycolysis must be given to NADH or FADH2. Those shuttles I'll make a separate video, but I'm assuming that the the operating shuttle has given the electrons to the NADH. Okay, so now NADH is getting oxidized here. So complex 1, what is going to happen is at complex 1 the NADH is going to get oxidized. So NADH is going to get oxidized, NADH is going to get oxidized, and it is going to get converted into NAD plus. Okay, now when NAD plus is released, I told you it is carrying how many electrons? 2 electrons. So this NADH is oxidized NADH upon oxidation will give away its 2 electrons to first FMN. Okay, first it is going to give its electrons to FMN. So FMN is the first electron acceptor here. Okay, so from FMN the electrons are passed to iron sulfur center. Now this order is very important. Okay, from the iron sulfur center, the electrons are given to a compound called as ubiquinone. Okay, electrons are given to a small hydrophobic molecule which is called as ubiquinone. Okay, I will write it in short as UQ. Okay, when I'm writing the theory, I'll show you what is it. So this ubiquinone, before accepting the electrons, it is in the oxidized form. Okay, it is in the oxidized form. Now after accepting the electrons, okay, this ubiquinone will get converted into ubiquinol. Okay, so after accepting the electrons, it is going to get converted into ubiquinol. Now if you see here, most of the steps that are happening here are just reductions and oxidations. See, FMN got reduced, and from FMN, the reduced FMN got oxidized by giving its electrons to iron sulfur center. Now complex 1 got oxidized by giving away its electrons to ubiquinone. So the reactions that are taking place are just redox reactions. Okay, oxidation and reduction reactions are taking place here. So now I am telling you ubiquinone is reduced. Okay, UQH2. Okay, I am writing the reduced ubiquinone as UQH2. Now this is called ubiquinol. Okay, this is ubiquinol. I'll write it here itself so that there'll be no confusion later on. This is ubiquinone. Okay, this is ubiquinone. And remember, ubiquinone is a mobile electron carrier. Okay, ubiquinone is a mobile electron carrier. Okay, it's a mobile electron carrier. Okay, mobile electron carrier. It's a mobile electron carrier. It's a hydrophobic molecule. Okay, and it is a non-protein molecule. So three things about ubiquinone. Sorry for the orientation. I'll write the points about ubiquinone here. So ubiquinone is a hydrophobic molecule. It is a hydrophobic molecule. Ubiquinone is a hydrophobic molecule. Okay, and then it is a non-protein. It is not a protein. It is a non-protein molecule. Ubiquinone is a non-protein molecule, and this ubiquinone is also called as CoQ. Okay, it is also called as what? CoQ. It's also called coenzyme Q. Okay, so this is about the ubiquinone. Now let us come to complex 2. The complex 2 is called as succinate dehydrogenase complex. Okay, so what is complex 2 called as? The name of the complex 2 is succinate dehydrogenase complex. The name of the complex 2 is, it is called succinate dehydrogenase complex. It is succinate dehydrogenase complex. Succinate dehydrogenase complex. Okay, so I told you dehydrogenases are going to remove hydrogen, that is 2 protons and 2 electrons. Now this succinate dehydrogenase complex is going to oxidize FADH2, or FADH2 is oxidized at complex 2. Remember, FADH2 will never get oxidized at complex 1. So complex 1 oxidizes NADH and FADH2 is oxidized by complex 2. Now in this complex 2, okay, we have an iron sulfur center. In this complex 2 as well. Okay, now the electrons that are coming from FADH2, 2 electrons, okay, 2 electrons are first given to this iron sulfur center. From this iron sulfur center, again the electrons are given to the oxidized ubiquinone. So remember one important point. The important point is oxidized ubiquinone is going to receive electrons from both complex 1 and complex 2. So complex 1 as well as complex 2 are going to give their electrons to ubiquinone oxidized. Okay, so oxidized ubiquinone is going to give its it's going to take electrons from complex 1 also and complex 2 also. Okay, now these two complexes give their electrons to oxidized ubiquinone. Now what is important here? Now when two electrons are passing from the complex 1 to ubiquinone, okay, now let me take this as the matrix side. Okay, let me take this as the mitochondrial matrix. Okay, let me take this as this portion or this side of the membrane as mitochondrial matrix. Okay, so let me take this portion of the membrane to be as what? Okay, so this portion is mitochondrial matrix, and this portion is the inner membrane space. Okay, so the intermembrane space. So this portion, let us take it to be intermembrane space. Okay, so this portion is intermembrane space. Okay, intermembrane space. If you see the mitochondria, this is the, we are here now, this is the intermembrane space and this is the matrix. Now all these proteins are present here. All the proteins are here, on the proteins are present on the membrane. Okay, now so when for every two electrons that are passing, okay, from the NADH to, from the complex 1 to ubiquinone, okay, remember four protons, I'll just use a different color here. Four protons will move from the mitochondrial matrix. Four protons will move from the mitochondrial matrix. Okay, four protons will move from the mitochondrial matrix into the intermembrane space. Okay, four protons. So remember this. Four protons are moving from where to where? From the mitochondrial matrix into the intermembrane space. Okay, now let us move forward. Now what is going to happen? Let us come to complex 3. Complex 3 is called as cytochrome. Complex 3 is called cytochrome Bc1 complex. Okay, cytochrome Bc1 complex. Now why is it called cytochrome Bc1 complex? Okay, these are cytochrome B and cytochrome C1. Okay, so it is easy to remember. C is followed by B, or C comes after B, so the order is also like that. Cytochrome Bc1 complex. Okay, so the name of remembering it is also easy. Now this complex, which is called as, the name of the complex is cytochrome Bc1 complex. Okay, cytochrome Bc1 complex. Now this cytochrome Bc1 complex is going to, the electrons from the reduced ubiquinone or ubiquinol are going to be delivered to, or going to be transferred to complex 3. Okay, they're given to complex 3, and who's going to get the electrons first? They're given to whom? They're given to cytochrome B. Okay, here also you have an iron sulfur center. Okay, so cytochrome B, from cytochrome B to cytochrome C1. Okay, so the electrons are transported to cytochrome B and C1. Now from cytochrome BC1 complex, the electrons are given to, again one more compound, which is called as cytochrome C, which is called as what? Which is called as cytochrome C. Now from cytochrome C1, the electrons are transferred to cytochrome C. Okay, two electrons. Electrons are transferred to what? Cytochrome C. Cytochrome C is towards the which side? Is it towards the inner membrane side, or is it towards the matrix side? Cytochrome C is towards the inner membrane side. Remember that. Okay, cytochrome C is in the intermembrane space of the, is towards the intermembrane space side of the inner membrane of the mitochondria. Okay, now this cytochrome C is a mobile electron carrier. Okay, it's a mobile electron carrier between complex 3. This is complex 3. okay, between complex 3 and between complex 4. So cytochrome C is a mobile electron carrier between complex 3 and complex 4. See, questions can come as it is like this only. Question is going to be, which of the following is a mobile electron carrier between complex 3 and complex 4. Okay, the options can be plastocyanin. Remember, plastocyanin is also a mobile electron carrier. Okay, ubiquinone also mobile electron carrier. Cytochrome C also mobile electron carrier. Okay, so copper containing mobile electron carrier is different again. We know plastocyanin is a copper containing mobile electron carrier. Okay, so all that we'll study later. So cytochrome C. Now from cytochrome C is mobile electron carrier. Now from cytochrome C, the electrons are passed to, okay, the fourth complex, complex 4, which is the cytochrome A3 complex. Okay, what is it? Cytochrome A3 complex. Okay, cytochrome A3, or you can write it in short as cytochrome A3 complex. Okay, or some books also write it as cytochrome C oxidase complex. Okay, some books also write it as what? Cytochrome C oxidase complex. Okay, cytochrome C oxidase complex. Cytochrome C oxidase complex. Now, why is it called cytochrome C oxidase complex? See, it is oxidizing cytochrome C and taking electrons from cytochrome C, correct? So, because it is oxidizing cytochrome C and taking electrons from cytochrome C, it is called as cytochrome C oxidase complex, or you can also call it cytochrome AA3 complex. So this is the fourth complex, and this is the last complex of the electron transport chain. Now here the electrons are first taken by cytochrome A, okay, cytochrome A followed by cytochrome A3. Actually this is copper containing. Copper is also present in this. Okay, copper is also present in this. So cytochrome A3. So copper is also present in this fourth complex, that is cytochrome A3 complex. Okay, now finally the electrons from here, that is cytochrome AA3 complex, finally the electrons from here are given to electrons from here are given to the two electrons from here are accepted by oxygen. Are accepted by whom? Oxygen. So two protons plus, okay, half O2 will accept two electrons, and it is going to form a water molecule. It's going to form what? It's going to form a water molecule here. Okay, water molecule. Now this one is called as the ATP synthase. This is called as ATP synthase. Okay, this is called ATP synthase. Okay, so the final electron acceptor, again a famous question. Final electron acceptor is oxygen. Final electron acceptor is oxygen. First electron acceptor, FMN, complex 1 took the electron if it is NADH. Okay, now for every two electrons passing from the for every two electrons passing from complex 3 for every two electrons passing from complex 3, okay, again four protons are pumped into the intermembrane space. Here for complex 4, for every two electrons passing from complex 4, two protons are pumped into where? Two protons are pumped into the intermembrane space. Okay, so if you see the overall number of protons that are pumped into the intermembrane space from complex 1, how many protons are pumped? Four protons are pumped from complex 1. From complex 3, how many protons are pumped? Again four protons are pumped. From complex 4, how many protons are pumped? Again two protons are pumped. So if one single NADH2 molecule or one single NADH molecule is getting oxidized, how many protons are pumped? So 4 plus 4, 8 plus 2, 10 protons are pumped into the intermembrane space. So this is very, very important. You should remember this. Okay, so complex 1 is pumping 4, complex 2 is pumping 4. Sorry, complex 2 doesn't pump any protons, complex 3 is pumping 4, complex 4 is pumping 2. Okay, so 4, complex 1 is pumping 4, 1, 4, 3, 4 and 2, 4. Sorry, 4, 2. 4 is 2. Now, 4 is 2. Now what about complex 2? Complex 2 doesn't pump any protons, okay? Complex 2 doesn't pump any protons from the mitochondrial matrix into the intermembrane space, okay? It doesn't pump any protons, so bear this in mind. If there is, out of the 4 complexes, which complex doesn't pump protons? It is complex 2, okay? Now if you see this redox reactions, now how can the questions come from here? What are the questions possible from here? Okay, so what questions are possible from here? First and the foremost thing is the order. Okay, order of the electron movement, it's very important. Okay, order of the electron movement is very important. Second thing is which one is getting oxidized, which one is getting reduced. Okay, which one is getting oxidized, which one is getting reduced. The third thing is they can ask which one is more electronegative, which one is less electronegative. You know that electrons are taken by a more electronegative atom and electrons are given away by the less electronegative atom. So that is also important. So based on the movement of electrons, you can say that whichever is gaining electron, for example, oxygen is the final electron acceptor. So oxygen is the most electronegative atom, correct? I mean, fluorine is still there, but in the living systems there's no, we don't fluorine is not in the biological systems. You don't find fluorine at many places. So oxygen is the most electronegative atom here. Okay, so these are the areas from which from electron transport chain questions can come. Apart from this, energy calculations are there. Energy calculations, I'll make it a separate video. Electron transport chain is not over now. We have just seen what is happening here. Now the key and the important concept of electron transport chain is the proton accumulation, okay, or accumulation of protons in the intermembrane space. Okay, so what are we creating? We are creating a gradient, we are creating a difference, or this electron transport system is creating a difference in what? Difference in protons between what and what? Difference in protons between the mitochondrial matrix and the intermembrane space. Okay, it is creating a, see here, I'll just show it here. Now we are here. Okay, now this is the mitochondria, correct? This is the mitochondria. Let us assume that this is a mitochondria. Okay, and this is the intermembrane space. Okay, this is the intermembrane space. Now from the matrix through these electron transporters, protons are being pumped here. Four protons, 2 protons are being pumped here. So what is happening in the intermembrane space? In the intermembrane space, the proton concentration is increasing. In the intermembrane space, proton concentration is increasing, means acidity is increasing. What is increasing? Acidity is increasing, means pH is decreasing. And who's responsible for this? Electron transport chain is responsible for this. So electron transport chain has created a gradient. It's created a difference. Okay, gradient means difference between what and what? Between the difference in protons, or different in the protons between the mitochondrial matrix and the intermembrane space. Now where are the number of protons more? The number of protons are more in the mitochondrial matrix, intermembrane space. Intermembrane space of the mitochondria, we have more number of protons in comparison with the matrix. So this gradient is created. Who created this gradient? It was created by the electron transport chain and the protein complexes that are arranged. They are responsible for it. Now what is the use of creating this? Difference or gradient? Okay, that we will see in the next video. Okay, so that video is going to be electron transport chain 2. Okay, where we will see how this proton gradient that is created is going to help us in generating ATP. This is the main drama here. This is, now we have set the stage. For, okay, creating ATP. We have set the stage for making ATP from ADP. We have created the stage for making ATP from ADP. Okay, now how this ADP, okay, is getting converted into an inorganic, with the addition of inorganic phosphate is getting converted into ATP is what we are going to see in the electron transport chain video 2. Okay, so till then revise this concept once from NCERT very thoroughly. Remember the order of the complexes. Okay, the order of the complexes, names of the complexes can be a match matrix, and movement of electrons also can be a match matrix. Okay, so I'll just write it here. I'll just write the moment of electrons here. So NADH, from NADH the electrons are passed to FMN. If you want you can copy it down. FMN to the iron sulfur center. From the iron sulfur center it is going to ubiquinone. Ubiquinone, I'm writing it as UQ. Okay, from UQ it is going to complex 2, that is cytochrome B. From cytochrome B to cytochrome C. From cytochrome C to it is going to BC1 complex, C1. From cytochrome B to cytochrome C1 to cytochrome C, and from cytochrome C, okay, from ubiquinone to cytochrome B to cytochrome C1, from cytochrome C1 to cytochrome C, from cytochrome C it is transferred to cytochrome A. Cytochrome A, from cytochrome A it is transferred to cytochrome A3. Okay, this is complex 4. From cytochrome A3 it is finally accepted by oxygen. Okay, oxygen, these two electrons are finally accepted by oxygen. These two electrons can be either from NADH or from FADH2. Okay, so plus half oxygen plus these two electrons, okay, will give rise to a water molecule. Okay, they're going to give rise to a water molecule. Okay, so this is also very important. Remember, this is also very important. This is also very important in the sense sometimes how many electrons are required to make 2 molecules of oxygen, or how many electrons are required to make 4 molecules of oxygen. Such questions can come, so we will do those questions also. Okay, so this is so far the stage that is set. Okay, now we have to do chemiosmotic hypothesis, or that is making of ATP, that we'll be seeing in the next video. Okay, thank you very much. And if there is any doubt, or if you have any issue with any concept that we have discussed, you can write it down in the comment box. I'll answer them. Thank you.